Hello everyone, and welcome back to this lecture where we are learning about DNA replication. In the last part, I explained that DNA replication is semi-conservative, semi-discontinuous, and it occurs bidirectionally. In this part, I will focus on the proteins and enzymes involved in DNA replication before moving on to continue with our overview of gene expression. DNA replication in E. coli is a highly coordinated process. At a replication fork, two polymerases are active, one synthesizing the leading strand and the other synthesizing the lagging strand in Okazaki fragments. The two DNA polymerase enzymes each act as a complex of multiple subunits in order to synthesize DNA. We know that for this to happen, the double-stranded DNA needs to unwind from the origin of replication to form a replication bubble. There are several proteins and enzymes that are involved in this process. A DNA helicase uses the energy from ATP to unwind DNA, and a topoisomerase helps by relieving the strain that results in the downstream DNA. Single-strand binding proteins prevent the unwound DNA from renaturing. Before DNA synthesis can begin, though, we know that DNA polymerase needs a primer since it cannot start a new strand from scratch. I explained before that the enzyme called primase synthesizes an RNA primer from which DNA can be elongated. As illustrated here, primase associates with the helicase where the fork splits in what is called a primosome. In this slightly simplified illustration, we see that the DNA polymerase that continuously synthesizes the leading strand does so in the 5' to 3' direction towards the fork, and the polymerase that discontinuously synthesizes the lagging strand does so in the 5' to 3' direction away from the fork. Yet evidence suggests that the two DNA polymerases associate together in a single complex while synthesizing DNA. So how do we reconcile this? We can imagine that with the two copies of DNA polymerase associated together, the template of the lagging strand loops back around to allow the polymerase that acts discontinuously to synthesize an Okazaki fragment. So we see that in addition to DNA polymerase, there are many other key proteins and enzymes involved in DNA replication. Looking at DNA polymerase itself, we see that in E. coli there are actually three kinds of DNA polymerase that are active: DNA polymerase 1, 2, and 3. DNA polymerase 3 is the main enzyme involved in DNA replication, and it is the one illustrated in the figure here as two copies synthesizing leading and lagging strands of DNA, elongating them from RNA primers. DNA polymerase 1 plays an important role in removing the RNA primers and replacing them with DNA, as we discussed in the previous part of the lecture. It can also act in DNA repair, a function that is also carried out by DNA polymerase 2. There are also multiple DNA polymerases found in eukaryotes, which I won't discuss here, but details can be found in Table 20.2 of your textbook. In the previous part of the lecture, we had already discussed the role of primase in synthesizing RNA primers, which allow DNA polymerase 3 to start replicating DNA. And we also explained that DNA ligase joins Okazaki fragments together once the DNA has been replicated. Here I have just introduced several other proteins. Helicase separates the parental strands of DNA using the energy from ATP. Single-strand DNA binding proteins stabilize single-stranded DNA, preventing reannealing of the strands separated by helicase. And topoisomerases relieve the torsional strain on DNA that results from unwinding. Helicase binds and moves along DNA, separating annealed strands as it goes. Helicase has to separate the DNA at physiological temperature, so to unwind the double helix below the melting temperature, it hydrolyzes ATP to provide the energy required to pull apart the strands. Single-strand DNA binding protein cannot catalyze strand separation, but it selectively binds single-stranded DNA, forming a coat around it and preventing reannealing of separated strands. As DNA strands are separated, this causes strain downstream of the fork. Since DNA is a double helix, strands must rotate around each other when they separate, and this results in supercoiling. You could imagine this if you twist two bands together and then pull them apart, as illustrated in this figure. As the strands supercoil, this results in more resistance and the replication fork will stall. To release the tension, a topoisomerase will transiently cut the DNA either in one or both strands, untwist, and then reconnect it, relieving the strain by changing the DNA topology. Of course, the main player in DNA replication is DNA polymerase. This enzyme was discovered from E. coli by Arthur Kornberg in 1958. To polymerize DNA, the enzyme requires a template strand, a strand to elongate, and deoxyribonucleoside triphosphates, or DNTPs. DNA polymerases have a highly conserved structure. The first crystal structure of a DNA polymerase in complex with double-stranded DNA was solved in the 1990s and is shown here in figure 20.16. The structure of the enzyme forms a cleft between two helices made up of what can be described as palm, thumb, and finger domains. The DNA binding site is within the cleft, sitting at the palm. We know that DNA polymerase III is the main replicative enzyme in E. coli, and that DNA polymerase I and II are involved in DNA repair. Remember also that DNA polymerase I plays a key role to remove the RNA from Okazaki fragments, using its 5' to 3' exonuclease activity to digest the RNA before replacing it with DNA. Both DNA polymerase I and DNA polymerase III have proofreading capability, which results from a 3' to 5' exonuclease activity. The result of this 3' to 5' exonuclease activity is increased fidelity. When the polymerase adds a base that is mismatched to the template, the incorrect nucleotide at the 3' end of the growing strand can be excised before DNA synthesis continues. In effect, this provides a proofreading mechanism for the enzyme to go back and correct a mistake it has just made before moving onwards. Without this proofreading mechanism, the error rate of DNA polymerase III is about 1 in 100,000, but with the 3' to 5' exonuclease activity, the error rate is reduced to 1 in 10 billion. So, coming back to all of these things working together in the highly coordinated process of DNA replication, we have DNA first unwinding at the origin of replication to form a replication bubble. Helicase pulls DNA strands apart using the energy from ATP, and single-strand binding proteins prevent the renaturation of denatured strands. The resulting torsional strain in downstream DNA is relieved by topoisomerases. Primase, which is associated with helicase in a primosome, synthesizes RNA primers allowing the DNA replication to start. As the replication bubble then expands with replication forks moving outwards, DNA polymerase III replicates DNA in leading and lagging strands, with synthesis being continuous in the leading strand and discontinuous in the lagging strand, resulting in Okazaki fragments. To complete replication, DNA polymerase I replaces the RNA primers with DNA sequence, and DNA ligase joins Okazaki fragments together. To reiterate all of this, we've learned that DNA replication is semi-conservative, and replicated daughter DNA contains one new strand and one parental strand. DNA replication is semi-discontinuous, with the leading strand synthesized continuously and the lagging strand synthesized discontinuously as Okazaki fragments. Replication of a DNA replicon begins at an origin of replication and proceeds through a coordinated process involving multiple proteins and enzymes. And we have learned that DNA is synthesized bidirectionally, although there are cases in nature where it can be synthesized unidirectionally. Moving on from DNA replication, we'll continue our overview of gene expression, focusing next on transcription of DNA into RNA. The idea that DNA needs to first be transcribed into RNA before the sequence can be translated into protein was first proposed by Francis Crick, based on the fact that in eukaryotes, DNA is in the nucleus, but protein synthesis occurs in the cytoplasm. Information on DNA coding for proteins needs to be carried out of the nucleus and into the cytoplasm, where protein synthesis is carried out by ribosomes. But there are several types of RNA in the cell, so which one carries the message? The ribosome is the protein synthesizing machinery of the cell. It is a complex of mostly RNA and some protein, and Francis Crick thought that the RNA that forms the core subunits of the ribosome carried the coding message for proteins, and therefore that each ribosome is dedicated to synthesizing a specific protein. Francois Jacob, on the other hand, proposed an alternative hypothesis that the coding message was carried by a transient RNA molecule that was not permanently associated with the ribosome. To distinguish between the scenarios proposed by these two hypotheses, Jacob, Brenner, and Meselson carried out experiments using isotopic labeling. They found that a transient messenger RNA, or mRNA, that is distinct from ribosomal RNA, or rRNA, was the information carrier. In performing their experiments, these researchers used T2 phage, which is a virus that takes over E. coli's protein synthesizing machinery, providing instructions to make new proteins. In order to trace the phage RNA coding for viral proteins, they used both stable and radioactive isotopes to label ribosomes and RNA. They first grew E. coli in a medium containing stable nitrogen-15 and carbon-13 to label ribosomes before phage infection with heavy isotopes. Then, they infected the cells with the T2 phage and switched the medium to one containing light isotopes, nitrogen-14 and carbon-12, so that the newly synthesized ribosomes would have lower density, and at the same time they added radioactive phosphorus-32 to label and trace phage RNA. If the information coding protein synthesis was fixed in ribosomes as part of ribosomal RNA, then they should have seen radioactivity associated with the newly synthesized lower density ribosomes, which could be distinguished from older higher density ribosomes by density gradient ultracentrifugation. In the hypothetical scenario where protein coding information is hardwired into ribosomes on ribosomal RNA, they expected there would be no radioactivity associated with the old higher density ribosomes. Alternatively, if the information coding protein synthesis was on a transient messenger RNA, then they would see it associate with the old ribosomes that have higher density. In fact, this was the result that they observed after infection with phage. The new phage RNA was seen on the old ribosomes, and infected bacteria did not produce any new ribosomes, but instead spent all of their resources synthesizing viral proteins. And so Jacob, Brenner, and Meselson concluded that mRNA is distinct from the ribosomal RNA that makes up the ribosome. Their results implied that the same ribosomes, made up of rRNA and protein components, can synthesize different proteins. The message coding for proteins is carried on mRNA, which is short-lived and transiently associates with the ribosome during protein synthesis. So we have established that RNA carries the information from genes coding proteins to ribosomes, the protein synthesizing machinery. But how is RNA produced? Protein coding RNAs and other kinds of RNA are synthesized by enzymes called RNA polymerases. RNA is polymerized from ribonucleoside triphosphates as precursors, with RNA polymerase catalyzing the synthesis of a phosphodiester bond to each newly added nucleotide with the release of diphosphate, as shown in this figure here using the notation of your textbook. If the figure is not clear, it can also be represented like this. While a diphosphate is released from each incoming ribonucleoside triphosphate that adds a nucleotide to RNA, you may notice that the first nucleotide at the 5' end retains 3 phosphate groups. Of course, the synthesis of RNA is normally templated. I've drawn the template here with complementary bases. I might ask you, what is the polarity of this template? Is this end here the 5' end or the 3' end? The templated synthesis is shown here in this figure using the notation of your textbook. In transcription, the template is DNA, but RNA can act as a template in RNA replication. Transcription can be divided into 3 stages. The first stage is initiation, when RNA polymerase binds to the double-stranded DNA at a specific sequence called the promoter. It melts or unwinds a short stretch of DNA, and the first few nucleotides are added to a newly synthesized RNA molecule through phosphodiester bonds. The second stage is elongation, when the RNA polymerase starts to move along the DNA template, with the melted, unwound bubble of DNA moving along with it, allowing the RNA polymerase to read the bases along the DNA template and continue to add more nucleotides to the growing RNA. The third and final stage is termination. This occurs when the RNA polymerase reaches a termination signal in the DNA, causing the polymerase to stop transcribing and fall off of the template. To summarize our overview of transcription, in transcription, a sequence of nucleotides is copied from DNA into RNA. This is catalyzed by an RNA polymerase using a DNA template. One strand of the DNA is the template strand. This is also called the antisense strand. The non-template strand of DNA has the same sequence as the transcribed RNA, and is also called the sense strand because it has the same sense as the RNA. Now we've talked about both DNA replication and transcription of DNA into RNA. It's worthwhile to talk about some of the similarities and differences in both processes. In DNA replication, DNA is synthesized using a DNA template. In transcription, RNA is synthesized using a DNA template. In DNA replication, the replication machinery is recruited to an origin of replication. In transcription, the transcription machinery is recruited to a gene promoter. In both DNA replication and transcription, the newly synthesized strand is polymerized from the 5' end to the 3' end. In DNA replication, the polymerase requires a primer, whereas in transcription, RNA polymerase can initiate RNA synthesis without a primer. In DNA replication, both strands are copied, whereas in transcription, only one strand is transcribed. And of course, in DNA replication, T's are added where they base pair with A's in the template, and in transcription, U's are added where they base pair with A's in the template. That completes our brief overview of transcription in this lecture. Transcription is a highly regulated and coordinated process, which we will come back to in excruciating detail in chapters 6 to 12, where we will discuss specific aspects that control transcription in both bacteria and in eukaryotes. But for now, that's the end of this lecture.